Another type of vectors which are in use in rDNA technology are artificial chromosome vectors. So they are artificial and they are chromosomal vectors. So different types of artificial chromosome vectors we have. For example, bacterial artificial chromosomes, abbreviated as BAC. Then we have PAC. Actually, they are P1 bacteriophage based chromosome vectors which are derived from E. coli. YAC, that is yeast artificial chromosome, and we have HAC, that is human artificial chromosome. Now we will see one by one what is their use in or how they can be a vector of choice in rDNA technology. BAC, it's a bacterial artificial chromosome. They are simply plasmids designed for cloning of large or long segments of DNA, so they can take up to 50 to 250 kilo base pair DNA fragment. They generally include selectable markers such as an antibiotic resistant gene. Here we can have a resistant gene for chloramphenicol, and also they will possess a stable origin of replication so that it maintains plasmids at 1 or 2 copies per host cell. Next we have PAC, that is P1 bacteriophage artificial chromosome, which is derived from E. coli. So they can also take up 50 to 250 kilobase pair DNA fragments, and both BAC and PAC they require E. coli as host. Then we have YAC, that is yeast artificial chromosome. So the importance of this is they can take up a large segment of DNA, somewhere around 500 to 3000 kilo base pair, and they also have all elements which can maintain the eukaryotic chromosome in a yeast nucleus, like origin of replication, and also they will have 2 selectable markers which can be used for identification of these clones, and also they will have a specialized sequence needed for the stability of chromosomes during cell division. So even though they can take up large fragments, they are unstable and they are prone for rearrangements and difficult to separate from the endogenous yeast chromosome DNA. So that's why BAC and PAC are preferred over YAC, because BAC and PAC they can take up to 250, and also their molecules can be easily separated from the E. coli genomic DNA using standard plasmid isolation. So because of these two factors, so they can take up a large fragment, and also they are very stable, makes BAC and PAC a preferred one when we compare it to YAC. Then we have HAC. So HAC stands for human artificial chromosome. They are actually useful in expression studies. So whenever we want the gene to be expressed, it's better we go with this cultured or artificially structured human chromosome. So they will have, instead of 46, the 47th chromosome, which can carry a new gene or a desired gene. And these HSCs usually require cultured human cells as their host. And the important feature is that these HSCs can be used whenever we want to have transgenic animals, and also they are very much used in gene therapy. Expression vectors. So expression vectors are the vectors, they not only amplify in the host or propagate in the host, but also they help in expression of the cloned gene. So the foreign DNA, whatever it is inserted into the vector, is expressed to produce the product. So the product is the protein. So these type of vectors are called as expression vectors. So these expression vectors will have all the machineries or required sequences for transcription and translational process. So these expression vectors are used whenever we want a production of proteins or the expression of gene. For example, one of the utility of these expression vectors are if we want to produce a hormone like insulin. So we can go with expression vectors. When we have so many vectors in hand, how to choose or which one to choose for the experiment? So when this question arises, the choice of vector mainly depends on the DNA insert size. So what is the size of the desired DNA or the gene which we want to insert into the vector? That will tell which one we have to choose for the experiment. For example, if you want to insert a DNA of very small size up to 10 kilobase pair, we can go with plasmids. Like that, if it is little bit higher size, bacteriophage lambda, and cosmids can take up to 35 to 50 kilobase, and bacterial artificial chromosomes can go up to 250 kilobases, and yeast artificial chromosomes can take up to 3000 kilobases. So it is the DNA insert size which makes us to choose which vector for the experiment.